Hello, this is Group 2, and for our project, we'll be discussing CRISPR technologies and the 2015 moratorium. So CRISPR is short for clustered interspaced short palindromic repeats, and it describes the genetic appearance of a system. This method functions almost like an immune system used by microbes to protect themselves from harmful viruses. CRISPR establishes a genetic memory and creates a guide RNA sequence to find viruses through the complementary sequence. So that the next time that bacteria infects the cell, RNA will be able to recognize, bind to, and then destroy the sequence. Cas9 is a complex created by two RNAs with a protein. Cas9 is a nuclease or a type of enzyme that can cut DNA. So when a complementary sequence or a guide RNA acquires a target within a viral genome, that DNA can be cut so the virus is then disabled. This same cut and paste system can be engineered to cut DNA at any given location. So Cas9 can be utilized to target and alter human genomes in sperm and egg cells, also referred to as germline cells. It's very important to note that these alterations can be done within a living cell. And in the bigger picture, this provides a unique opportunity to alter the human race. This quickly became very worrisome because moving forward, this allows for targeted genotypical and phenotypical mutations. Thus, a moratorium was called starting in 2015. It's not new to scientists to know how to modify genes. Not only that, but it was formerly a more lengthy, difficult, and expensive process. However, the use of CRISPR and Cas9 technology streamlines gene editing and makes it more accessible. While the possibility to make precise changes to genomes is exciting, it opens the door for editing and prioritizing specific genetic traits such as height, hair color, intelligence, etc. Naturally, grew concerns for creating quote unquote designer babies and the possibility of engineering the human race moving forward. There were divisions among scientists on how to proceed, but a containment strategy was proposed by several groups of scientists and bioethicists to raise awareness for their concerns of these advancements. The intention was to cultivate trust and ideate ways of proceeding judiciously, and this started in 2015. That same year, the National Institute of Health, or the NIH, announced that it would not be funding germline editing on human embryos, and Congress passed the first of many bans of altering germline cells intended for pregnancies. Between 2016 and 2018, national scientific associations, as well as research groups, provided guidelines for genome editing for its other applications, such as in clinical trial treatments or even for the treatment and even removal of disease. And I say disease in quotes, depending on how one defines that. By 2019, this topic had become internationally controversial as a global moratorium was called, and this was following a Chinese scientist or a group of Chinese scientists who had genetically edited embryos, a couple of which were even carried to term. In response, US senators swiftly denounced this and called for the construction of international guidelines. However, at around the same time, some groups began advocating for a partial lift of the ban and changes to restrictions for the use of CRISPR and Cas9 therapies, such to include other, gene therapies such as three-parent IVF. But in that same year, the NIH made its support for a 5-year international moratorium clear and stated that these experiments for clinical purposes were, quote, irresponsible and unethical. Since then, there hasn't been much movement regarding regulation, but the ethical concerns of using CRISPR and Cas9 technology has continued to grow.